these are notes on Mendelian genetics vocabulary, and they can be found in further detail on pages 492 to 495 in your book. So the term Mendelian genetics comes from this guy, Gregor Mendel. Gregor Mendel was a monk who lived in Austria. He was Austrian from 1822 to 1884. And in those times, monks, right, so he was a Catholic monk, they also did a lot of science. They had time and they had the resources of the church to really do scientific experiments. And so he experimented on pea plants. And so you can see in his picture here, there are some, beautiful pea plants in the background or the foreground, whatever. And you can see that there are different traits that he could study. So pea plants can have purple flowers or they can have white flowers. They can have yellow peas or they can have green peas. The peas themselves can be round or they can be shriveled. So lots of different traits within the same species of pea plant. And Mendel, in the same way that we pollinated our Brassica rapa plants in class so that we could see the trait of stem color being passed down, Mendel pollinated his pea plants and so that he could experiment on different traits and learned a lot about how traits are passed down through generations. And he's really considered the father of modern genetics. So you'll hear about Mendelian genetics if you're studying genetics, and that's where that comes from. So I wanted to give you some basic Mendelian vocabulary. So genotype, if we say genotype, those are the genes that you have. And so when we've been looking in the cell nucleus and we've been drawing the little genes on the chromosomes, that would be your genotype. And I'm going to give you examples of this in a minute and connect it to what we've already learned. Phenotype are the traits you express. So as it turns out, you could, and you should already know this a little, you could have different genotypes, different genes, and the same phenotype. So go back to like on your test, we looked at tasting PTC paper. You could have two genes that make functional taste receptors, or one gene that makes functional taste receptors and one that does not. But in both cases, you'll be able to taste PTC. So you would have the same phenotype, but different genotypes. Allele, some of you already asked about this from the protein reading. Allele is the form of a particular gene that you have. So again, back to PTC tasting, you have an allele for tasting PTC, an allele for not tasting PTC, and you could have any combination of two not tasting, two tasting, one and one. Those are both PTC tasting genes. They're just two different alleles, so two different forms of that gene. So now let's connect to what we've already learned in genes to traits. So here, this cell, I didn't actually say this specifically, so I'm going to physically say it. This cell is going to represent a freckle gene. So what happens in freckles is that there is an enzyme. Okay, so here we've got our little enzymes. Let's get our pen here. So here we've got our enzymes here and here. These are our functional enzymes. Here we've got some non-functional enzymes. So obviously this was the functional gene. This was the non-functional gene. Here we would say these are two alleles of the freckles gene, a functional allele and a non-functional allele. Remember, they're both making some kind of protein. It's just that the non-functional one is making a non-functional protein and the functional one is making a functional protein. And so as we may remember, these are going to represent just skin-colored pigment molecules, okay? We all make skin, well, not everybody. Some people are albino, but that's a different situation. But most of us make some kind of skin-colored pigment molecule, regardless of how dark or light that molecule may be. What a freckles gene is it's an enzyme that turns that skin-colored molecule into a darker pigment. So you can get freckles starting with any skin tone because your freckles molecule is going to be darker than whatever your personal skin tone is. And so, we've learned that, okay, these two go in and they make these darker colored pigment molecules. Now, what we've talked about is, regardless of the fact that we've got some non-functional enzyme here, because enzymes can be reused, this molecule here, if it goes into a non-functional one, can still later go into a functional one, and you can end up with freckles. So now let's connect this to our vocabulary. So the genotype, we're going to say, is big F, little f. So we're going to make our functional protein. We usually make genes different letters just because it's easy. So we're going to say this is big F, and we're going to say this is little f. Sometimes we have letters that make sense for the gene, and sometimes it has to do with the particular protein. So I'll show you an example later where it's a combination of letters that stands for the gene, and it has to do with the protein that's made there. So we would say that this genotype is big F, little f, because there's one functional and one non-functional, and we're always going to make the functional one capital. The phenotype is the person has freckles. The alleles in this person, there are two alleles. There is a functional freckle allele and a non-functional freckle allele. And then it's diploid because there are two freckle genes, two chromosomes that are making the freckle gene. But I do want you to remember, this is a very simplified model. I want you to remember that most traits are determined by more than one gene pair. So if you think back to fruit fly eye color, that's something like 8 different genes. Of those 8, so for those 8, there's 2 copies of each of those 8. So there would be like an enzyme 2 gene and an enzyme 3 gene, but there's 2 copies of the enzyme 2 and 2 copies of the enzyme 3. And I'm only saying that to let you know that most genes are a lot more complex, right? Freckles, you can kind of work your way through and say, oh yeah, I get it. There's one enzyme and it's going to turn skin color pigment darker. And if I have any of that enzyme, I'm going to have some darker pigment, so I'm going to have freckles. And you can work through that, right? PTC tasting is another one. It's a single gene trait. You can work through if I have any functioning PTC taste receptors on my tongue, I'm going to be able to taste this. You can't do the same thing for, obviously, fruit fly eye color. You can't say, well, if I have any functioning enzyme 3s, I'm going to have scarlet eyes because maybe I'll have wild type eyes. Maybe I'll have something else, right? I can't remember I'm trying to remember the pathway in my head. But the same thing's true for human eyes. You can't just say, if I have any copies of this, I'm going to have this color eyes, because it's more than one gene. So there's more than one possibility. So there's more than one combination. So now I want to talk about dominant versus recessive, that some of you have been using, whether or not you're using them correctly or not. You believe you're using them correctly. Some of you are not. So what dominant means is that one allele is needed for the trait to be expressed. So like going back to freckles or PTC tasting, as long as you have one allele making a tasting receptor or making darker pigment, you're going to have that trait. So we would call PTC tasting or freckles dominant because you only need one allele. Recessive is you need 2 alleles for the trait to be expressed. So if you don't have freckles, it's because you have 2 alleles that are not making freckles. If you don't taste PTC, you would have 2 alleles that are not making functional taste receptors. So we would say that it is dominant to have freckles. It is recessive to not have freckles. It is dominant to taste PTC. It is recessive to not taste PTC. These are traits where it's a yes or a no. Don't think that every time it's yes versus no, it's dominant versus recessive. So I'm going to give you some examples where that's not the case. So, for example, the presence of freckles is dominant. So if you have freckles, we're going to make that a big F. If you don't have freckles, we're going to make that a little f. The letters you choose are whatever letters you choose. I would encourage you, when you start to work with these letters, that you choose capitals and lower cases that are significantly different from one another, rather than just smaller versions of the capital. So like F's are good, G's are good, A's are good, B's are good, but like S's, I've seen a lot of students confuse, like, wait, was that a capital S or a lowercase s? So this is where you really need to be aware of capital versus lowercase. So you have three choices. You could have two dominant genes or two functional freckles genes. And so that would be big F, big F. You would have freckles. And we would call that genotype homozygous dominant. So the dominant comes from the fact that they're both functional genes. Homo means same. We see homo all over the place meaning same. We see it in homogenized milk. We don't say homogenized milk. We say homogenized. But it means that all the milk has been blended together, so it's not separating out into like the cream part and the milk part. We see homosexual, right? That's people who are attracted to the same sex. So homo means same. Zygous means zygote. And your zygotes are your sperm and your eggs. So remember, where you got these genes were sperm and egg. So it means the zygotes, the sperm and egg carrying these genes, had the same gene, which is how you ended up with two of the same. You could also be little f, so that would be no freckles. Neither of your genes are making the functional enzyme. So that's homozygous again. The zygotes were still carrying the same gene, but in that case they were carrying the recessive gene. So we call that homozygous recessive. And then you could obviously be big F, and as we've talked a lot, you would still have freckles, but we call that heterozygous. Hetero means different. And so your zygotes were carrying different genes. One was carrying a dominant gene, one was carrying a recessive gene. And so that's heterozygous. And so you see the homozygous dominant and the heterozygous both have the same phenotype. They both have freckles, but they have different genotypes. And so that's where you could have separate genotypes but the same phenotype, whereas the homozygous recessive is a different genotype and a different phenotype. One thing I want to say is a lot of people have been saying, oh, but will you have fewer freckles? Or, you know, that's a hard thing to say. You know, if you're heterozygous, you may have fewer freckles, but freckles also tend to be, in some cases, environmentally determined by the sun. So you may have someone with fewer freckles simply because they don't have exposure to the sun, you know, go back to the tanning. You may have someone with fewer freckles because they're heterozygous. So that's where we say the trait is having freckles or not. It's not how many freckles you have, because that may be determined by a lot of different things. And so we have the same trait, freckles, but different genotypes. So I do want to state right now that there's a common misconception that dominant traits are somehow better than, stronger, overpower, or more prevalent in society, etc., than recessive ones. This is wrong. This is absolutely wrong. Dominant traits just mean you only need one allele to see that trait. Doesn't mean it's better, doesn't mean it's stronger, doesn't mean that you're more likely to pass it on to your children. If you're heterozygous, you have a 50% chance of passing on the dominant trait and a 50% chance of passing on the recessive trait. So here are some examples. The allele for 5 fingers and the allele for lactose intolerance are both recessive, and yet the prevalent phenotype in society is that recessive phenotype. Most people have 5 fingers. Most people are lactose intolerant. Whereas the allele for freckles and the allele for dwarfism are dominant alleles. And yet most people don't have freckles. Most people don't have dwarfism. Yet those are dominant traits. So keep that in mind, that this idea of, you know, in everyday speak, dominant usually means like stronger, or I don't know the better, but definitely stronger. That doesn't necessarily mean it in genetics. It just means you only need one allele. And there's only one way to really know if something's dominant or not, and that's to go back to your previous unit of genes to traits and use your knowledge of protein synthesis to determine dominance. So we can look at lactose intolerance and say, okay, I know that there's a gene making lactase. If I have any genes making lactase, I'm going to be able to break down lactose. So that's got to be the dominant gene. The same with freckles. I know that if any genes are making this darker pigment, well, making the enzyme for darker pigment, then I'm going to have those darker little spots. So I'm going to have freckles. That's got to be dominant. There may be some you don't know, right? I don't know that you can use that knowledge to work through dwarfism. So sometimes you just might need somebody to tell you, hey, dwarfism's dominant. If you have any genes creating whatever protein it is that causes dwarfism, you're not going to be a full-heighted person.